Sorry for being late. Are there any questions? Oh, wait a minute, I gotta get my thumb drive. Where do I have my thumb drive? Oh, there it is. All right, I did some grading. We're going to be working on chapter 8, genetics. Any questions before we begin? I don't think I'm sharing my screen. Let me share my screen. Should be seeing my screen now. That's not right. Let's see if I've got that. We talked about plasmids last time. Is that correct? Anyone? All right, I'm just going to assume that was correct. Um, Transposons. Can somebody confirm you can hear me?
I can hear you.
Thank you. Uh, Transposons are segments of DNA that can move from one region of DNA to another. You can call these jumping genes. A transposon actually moves from one place in the genome to another place. It could move even to a different chromosome. Transposons always have a transposase gene. Blow this up. Come on. Right here, which is always part of the transposon. Transposon is just the region of the DNA that can move from one region to another. It also has inverted repeats on both ends. It's thought the inverted repeats help the DNA insert and exert from the chromosome. The insertion sequences allow the cutting and the resealing of DNA, meaning the transposase genes actually will cut the DNA right here and right there, and then it will cut the DNA chromosome in another location, and then the transposon will insert itself. And it's thought that these inverted repeats help the insertion and exertion. Complex transposons can carry other genes, like the TN5 transposon carries the kanamycin resistance gene. And usually the complex transposons have an extra gene or two near the center of the transposon, and then they have a transposon over here and a transposon over here, meaning it has sort of like two transposons. And the reason is it just needs a transposase gene, but it only needs one. But it needs these inverted repeats. And so if it has the a transposon over here and over here, it has the inverted repeats in the right position. Any question about transposons? Transposons are important because they can move from the chromosome and then into a plasmid. And then the plasmid is much more likely to be spread in the population of cells in a population, meaning plasmids are much more likely to be spread among cells than the chromosome itself. And so a transposon can move DNA from the chromosome to a plasmid and vice versa. But it's important because it can move, like the kanamycin resistance gene, into a plasmid, and then this kanamycin resistance gene is more likely to spread in the population because it's in a plasmid. Any question about any of that? All right, let's briefly talk about genes and evolution. Genetic variation, that's genetic differences in a species, ultimately results from mutation. If there is no genetic variation in the population, then the only way it can result is from mutation. An example of this was before 10,000 years ago, in the human population, we only had one allele for eye color, brown eyes. And then 10,000 years ago, we had a spontaneous mutation in Northern Europe, creating the blue-eyed allele. So genetic variation resulted from spontaneous mutation. You could argue that dogs have yellow eyes. Can people sometime in the future get yellow eyes by spontaneous mutation? And it's a possibility. I don't know. Once genetic variation results in the population, meaning there's genetic differences in the population, it can then spread by crossing over. This reshuffles the genes, like, for example, the blue-eyed allele was initially linked to the white or blonde hair allele, and that is now switched. The blue-eyed allele is now on with the brownish or blackish eye color, and that's because of crossing over. Transformation, conjugation, and transduction can then spread genetic variation in the population, where transformation, conjugation, and transduction can take the DNA out of one cell and then move it into a recombinant cell, another cell. Transposition increases all this frequency of genetic shuffling around, sort of like crossing over, simply because it can allow the DNA to move from mainly the chromosome into a plasmid as well as elsewhere in the population. All of this can drive evolution, genetic variation, and that will move in the direction that natural selection takes it, meaning evolution moves via natural selection. Any question about any of that? All right, that's it for chapter 8. Let's look at the syllabus. I think we're going to talk about chapter 13, but let me look. Yep, not far enough. Either chapter 13 or chapter 14. Chapter 14. There is no lab today, but I will be logging in for the first 15 minutes to answer any questions. All right, let's begin with chapter 14. In chapter 14, you should really pay attention to the terms, because if you learn the terms of chapter 14, you've learned half of the material in chapter 14. So chapter 14 is very term intensive. Let's go ahead and shut that one down. As soon as it shuts down, let's switch over. All right. Chapter 14, the principles of disease and epidemiology. Obviously know the terms. To know all the terms, you should really learn the objectives. 2, understand the relationship of the normal microbiota and its host. 3, understand the stages of disease development. And 4, understand what nosocomial infections are and how to control them. Any questions about what we're going to be covering? All right, the first couple of terms we're going to talk about: pathology, the study of disease; etiology, the study of the cause of a disease. For example, the etiological agent of the disease anthrax is Bacillus anthracis. Any questions about any of that? Pathogenesis, the development of a disease. Infection, colonization of the body by pathogens, or the presence of a microbe where it is not normally found. For example, all of us have E. coli in our gut, and that's the normal location for E. coli. But if it's in your inner ear or if it's in your lungs, it can cause disease because it's not normally found in your inner ear or in your lungs. Any question about any of that? Disease is an abnormal state in which the body is not functioning normally. Infectious diseases are caused by microbes. Obviously, not all disease are caused by microbes, but infectious diseases are. When we're talking about disease, you should come to realize that few microorganisms are pathogenic, meaning few of them can cause disease. Only a small percentage of all total microbial species can cause disease. Some microbes are even beneficial to the host, and we'll talk about some of those today. Any question about any of that? All right. The normal microbiota is a large community of microbes found in association with all animals and plants. These communities are known as the normal microbiota. So we talk about the human normal microbiota and the kitty cat normal microbiota as well. And the species, many of them will be the same, like Staphylococcus epidermidis is both on the skin of humans and cats. However, cats have one, a Bartonella henselae, which is a part of their normal microbiota, which is not a part of our normal microbiota. In fact, Bartonella henselae, when it gets on a human, it can cause disease. That disease is called cat scratch fever. The normal microbiota is only found in certain regions of the host, only certain regions of the body. Disruption of the normal microbiota and the host balance appears to be associated with disease and impaired immune function. For example, when you take antibiotics, it kills off the normal microbiota, not all of them, but a lot of it, in your gut. And that can cause disease. Usually it's slight, but you could get constipation or diarrhea just from taking the antibiotics. Anyone had that happen? I certainly have. Fortunately, the normal microbiota host balance comes into rebalance, and that disease is usually short-lived. If it happens at all, it doesn't always happen. The normal microbiota is found in and on the body. So obviously our skin, large communities on our skin, in the intestines and in the gut, large communities in there. In the urinary and reproductive systems, especially the lower urethra, both males and females, in the vagina, the mouth, of course, that's part of the gastrointestinal tract, the throat, large microbial communities in the eyes, in the nose. Examples of places where the microbiota is not found are internal tissues and organs such as the blood, the cerebrospinal fluid, the muscle, the nervous tissue, and the bone. Normally, these regions are, what's the word, aseptic, and there are not microbes in these regions. However, if we do have them in these regions, you usually have an infection, and that would be caused by normal, or not normal, but a bacteria or microbe in this region causing disease. You can have a transient microbiota. It differs from the normal microbiota in that normally when like Staphylococcus epidermidis gets on a baby's skin as soon as the baby is born, the Staphylococcus epidermidis will stay on the baby's skin for the baby's entire life. And normally the normal microbiota colonizes the host, and then you can find it for essentially the lifespan of the host. That differs from transient microbiota, where the microbe may be present for days, weeks, or months, instead of the normal microbiota, where the microbe lives with the host essentially for the entire host life, or most of it. Any question about any of that? I have a story about a transient microbiota, and that is my aunt had a Staphylococcus aureus skin infection, and she was receiving medication for it because it was a skin infection. And we went to visit her and my mother and I, and she insisted on hugging us, which was fine. But I then was sampling my skin at that time about once a term, and shortly after that incident, I had Staphylococcus aureus on my skin. It must have transferred from my aunt when she hugged me to my skin. And I said, geez, that's not good. I never had Staphylococcus aureus on my skin before. I must have picked it up from my aunt. And I said, I'm going to have to watch this because Staphylococcus aureus is a human pathogen. And people who have it growing on their skin, about 20% of Americans, they're much more likely to get a skin infection. Usually it's something minor like a boil, but it can be another skin infection, which can get worse. Even a boil can get worse. But um, at that time, I was sampling my skin about every 3 months, every term. And every term after that first term, I never saw Staphylococcus aureus on my skin ever again. So for me, it was a transient microbiota. My normal microbiota threw off Staphylococcus aureus, and I only had it that one time. It was about a week after I was visiting my aunt and she hugged me. And she probably thought, well, I'm under medication. It probably won't spread. But it did. Any questions about any of that? All right. The normal microbiota and the host. Humans have about 3 times 10 to the 13th human body cells, and humans have about 4 times 10 to the 13th bacteria cells, meaning we have more bacteria cells in and on us than we have human cells. Any question about any of that? And that begs to ask the question, are we more microbe than we are human? Don't let the answer freak you out too much. Because by numbers, we have more bacteria than we have human cells. Here you can see some bacteria, the orange cocci, which they're not really orange color. In this photograph, they're orange, but they are in the nasal epithelium. These right there, those are not bacilli, those are actually villi from the nasal epithelia. And the villi help move the, those are not cilia, they help move the mucus, which you can see right there. There are some bacteria, they're not quite spirochetes, although that one is, spirals in the stomach. That right there is villi in the stomach, so that's part of the normal mucosa of the stomach. But those are some bacteria in the stomach. And there you can see, let me blow this one up. Some bacteria in the gut. There's a rod. There's a coccobacillus, couple coccobacillus. Someplace in here I see a cocci, but I don't see it now. Um, that right there is partially digested food. And of course, that's the uh, intestinal lumen of the intestines. Anyway, some of the normal microbiota. We found normal human microbiota in some unusual places. We've known about it now for about a little over 10 years, 10 to 15 years. They found that the female bladder is not sterile, that there are normal microbiota in the female bladder. And I've never found a study where they've studied males, so I don't know the question. Do males have a normal microbiota inside their bladder? And I just don't know the answer because nobody's ever studied it in males. Now, males have a longer urethra, so it is possible that only females have normal microbiota in their bladder. Any question about any of that? So the urine is never sterile because, at least in females, there's normal microbiota in the bladder. Uh, Mother's milk is also not sterile. Even after it comes out of the breast, it's obviously not sterile. But inside the breast, mother's milk is not sterile. That inside the breast tissue, mothers or females have normal microbiota. And it's actually important for an infant that mother's milk supplies um, normal microbiota for the infant, meaning the mother actually supplies bacteria that is beneficial to the infant. Any question about any of that? This one I didn't quite understand why this came as a surprise because they found that the placenta has a normal microbiota. And why this surprises me that this surprised scientist when they discovered it was the placenta is open to the outside. The female reproductive system is open so that microbes can get in it. What is a little surprising is that the placenta has a unique normal microbiota, which is only found when the placenta is there, which obviously is only when the female is pregnant. And then when she gives birth, the placenta is also born and the afterbirth. And the placenta is gone, so the normal microbiota of the placenta is obviously gone. Any question about any of that? By a unique microbiota, I mean we find species in the placenta that we do not find in other places on the any humans, including the female. All right, there is a relationship between the normal microbiota and the host. The normal microbiota colonizes the host and will be with the host essentially for the lifespan of the host. The normal microbiota is important because it can engage in microbial antagonism with a human pathogen. The normal microbiota is also important because it can help the host besides microbial antagonism, like the normal microbiota of our gut helps us digest our food. The normal microbiota also helps us make certain vitamins, like vitamin K and one of the B vitamins. A significant proportion, it's not the majority, but a significant proportion of the vitamins are made by our normal microbiota in the gut. So the normal microbiota is important, and it's one of the ways it's important, besides obviously helping us digest our food and other things, It can help us defend ourselves from a human pathogen. And that we call microbial antagonism, competition between two species of bacteria or other microbe. The normal microbiota can prevent the growth of pathogens by occupying the niche the pathogen might occupy. On our skin, like my skin here, I have normal microbiota there. And if a pathogen like Staphylococcus aureus were to fall on my skin, it may not be able to get established because it'll fall on top of Staphylococcus epidermidis and then not get any resources or food from my skin. So it could occupy the niche the pathogen might occupy, and then there's no room for the pathogen. It can compete for resources. Even if it were to fall on my skin, where there's no Staphylococcus epidermidis, I have lots of Staphylococcus epidermidis, as well as Micrococcus luteus and some other microbes on my skin that can compete for resources. And if they deny Staphylococcus aureus enough resources so that Staphylococcus aureus cannot reproduce, it will not get established on my skin and will not go on, grow, and then form disease. So competition for resources can happen. The normal microbiota can also change the environment, making the environment less hospitable for the human pathogen. For example, the normal microbiota on our skin, in our mouth, in the vagina, tends to make the environment more acidic. And that can make the pH too low, too acidic, for the pathogen to get established. Any question about that? And if you don't know, the skin is acid because of our secretions, meaning slightly acidic, but the normal microbiota um, metabolize the secretions on our skin and make it even more acidic. The same thing in the mouth and in the vagina. And then lastly, the normal microbiota can produce bacteriocins, a protein that can prevent the growth of bacteria, especially pathogens. For example, E. coli can produce a bacteriocin, which will then inhibit the growth of Salmonella and Shigella. You can think of bacteriocins as a very narrow-acting antibiotic. It's much more narrow than any antibiotic because E. coli can only, E. coli bacteriocin can only inhibit Salmonella and Shigella. And I've often wondered why we don't use bacteriocins instead of antibiotics, because somebody who had, I don't know, Shigella infection, instead of taking an antibiotic, which would kill off, let's say you use a narrow antibiotic and it only kills off gram negatives, because Shigella is a gram negative, but it's going to kill off all the gram negatives in your, or most of the gram negatives, I should word it that way, in your gut. And we have mostly gram negatives, a lot of gram negatives in our gut. Obviously, E. coli and a whole bunch of other bacteria are gram negatives. And you want to take the antibiotics to get rid of the Shigella, but you're killing off a lot of normal microbiota in your gut as well. And it would be better to take a bactericin, which worked specifically against Shigella, and then it would also work against Salmonella, some strains of E. coli. And that would be much better because it wouldn't hit other gram negatives in your gut, like Enterobius faecium, Enterococcus as well. Of course, that's a cocci, not a gram negative bacilli, but still it's in your gut. Any question about bacteriocins? Oh, and I have found an article discussing that bacteriocins were being used experimentally to treat certain diseases in a patient. So they are being used today experimentally, but they're not used routinely. Any question about microbial antagonism? You can also consume microbes in a probiotic. The consumption of live microbes intended to exert a beneficial effect. For example, individuals who take yogurt with live bacteria in it consume the microbes as a probiotic, and then these individuals are much less likely to get a urethral infection. For females, they're also much less likely to get a bladder infection. Any question about any of that? All right. The relationship between a normal microbiota and the host is one of symbiosis, where two different organisms or populations are living together. The normal microbiota and the host are symbiotic, but they are specifically a commensalistic or a mutualistic symbiotic relationship. You can have a pathogen with a parasitic symbiotic relationship. A parasite is where one organism benefits at the expense of the other. Like Staphylococcus aureus growing on my aunt's skin was parasitic. It harmed her. The Staphylococcus aureus got a home, got a food from my aunt, and then it harmed her, doing disease. I don't remember exactly what it did. It wasn't a skin boil, it was a skin disease. So the parasite benefits, the host was did not benefit. The host was harmed. Uh, um, Normal microbiota can form a mutualistic symbiotic relationship or a commensalistic relationship. A mutualistic symbiotic relationship is where both organisms benefit. For example, microbes in our gut get a home, the gut, and they get food from us, and they can benefit us by helping us digest the food and then making that available to humans. Like for cows, for example, the normal microbes digest the grass and then make sugar available to the cow. So that benefits the cow and then benefits the normal microbiota. For humans, we have a normal microbiota that makes vitamin K, which benefits us because we do need the vitamin K. We can't make it ourselves. And then the microbe benefits because it gets a home and it gets food from us. Any questions about a symbiotic relationship, mutualistic or parasitic? A commensalistic relationship is where one organism benefits and the other is unaffected. It's neither benefited or harmed. A good example of a commensalistic organism is in each of our eyes, we have one nematode. And this nematode must eat bacteria or other microbes that fall into the eye because it doesn't harm the human. It does not eat human cells in the eye. And it gets a home, so it benefits, and the human is unaffected. No benefit, no loss. So it's a commensalistic relationship. This is a good example because there's no minute benefit. Many other normal microbiota living on the host, they do help us a little bit. Like I said, Staphylococcus epidermidis and Micrococcus luteus growing on our skin, it can have microbial antagonism with the human pathogen. And so that would be a benefit, even if it's only slight, even if it's only every once in a while. We still would benefit from that. Any question about any of that? All right. The normal microbiota does not normally cause disease. Normally, in a healthy individual, the normal microbiota does not cause disease. However, it can cause disease under certain circumstances. The normal microbiota, if it does cause disease, We call it an opportunistic microorganism, one which can cause disease at least under certain circumstances. The normal microbiota can cause disease in compromised patients with a compromised immune system. For example, AIDS patients can get an infection from Staphylococcus epidermidis or Micrococcus luteus because they have no immune system. And then the Staphylococcus epidermidis or Micrococcus luteus could grow where it should not or cause problems that it should not. An example of an opportunistic pathogen is E. coli. It's normally found in the gut, but if it's found elsewhere, like in the inner ear or in the lungs, it is an opportunistic pathogen that should not be growing there, and it will cause disease when it grows outside the gut. Staphylococcus aureus is normally found in the nose and the throat of about 50% of Americans. Usually Staphylococcus aureus does not cause a problem in the nose and the throat. On the skin, it's actually found in 20% of Americans, but it does tend to cause problems in the skin. Even the people who, 20% who have it, they're much more likely to get a skin disease or a skin boil, which I guess a skin boil is skin disease, but any question about any of that? All right. I think this is the third time we've talked about Koch's postulate. If it isn't, then we'll talk about it one more time, but this might be the third time we've talked about it. Koch's postulate is the gold standard for identifying the cause of a disease with an infectious agent. Robert Koch first used the Koch's postulate to show that Bacillus anthracis was the cause of the disease anthrax. And this is the first time any microbe was shown to cause disease. Any question about any of that? All right, let's talk about Koch's postulate in a little more detail. If you can use Koch's postulates to show that a given microbe causes a given disease, it is advised you use it because it proves that this given microbe causes disease. What you do is you find a sick individual, isolate microbes growing in that individual, and then you have to isolate it and purify them, grow them up in pure culture, and then take that pure culture, inoculate it into a healthy individual, in this case, a healthy bunny. If that bunny then becomes sick, that's why the bunny's on its side. Ah, the poor bunny, it's sick. It's still not enough to say that this microbe causes this disease. You then have to show that you can isolate this microbe from this sick bunny. And if you have all of these, you can say that this microbe causes this disease. And that's Koch's postulate. Any question about any of that? Now, you should know that there are exceptions to Koch's postulate. What do you do if the microbe has a unique culture requirement? It cannot be grown in tissue culture. For many years, almost 100 years, scientists were never able to grow the disease which causes, just a minute, I'll get it, syphilis. And yet we knew which microbe caused syphilis. Another one, which I don't know if we can actually grow this one, syphilis, they've finally figured out what you need to add to a general purpose media to get syphilis to grow, meaning they finally figured out what to add. And it's a number of things, but they found it out. So in the last 10 years or so, we've actually been able to get syphilis to grow in the in the tissue culture. But long before we knew how to get syphilis to grow in the tissue culture, we didn't know which organism caused the disease syphilis, what bacteria. And Bartonella henselae is another microbe that for many years, maybe still today, we cannot get it to grow in tissue culture. We don't know what it requires to grow in tissue culture. And like I said, they may have figured that one out too. I don't know, but um, for many years, we knew what microbe caused cat scratch fever, Bartonella henselae, but for many years we could not get it to grow. So how did scientists figure out that these microbes that they could not grow in tissue culture, so they could not do Koch's postulates because you need to grow it in tissue culture to do Koch's postulates? How did we know that these microbes cause disease? Anyone have a guess? Nobody has a guess. Well, how do we know that HIV causes the disease AIDS? Anyone know that? Anyone have a guess? I'll give you extra credit on this one. All right. What you do is you study large numbers of the population, follow them over time, meaning you're studying them over time. And what you do is you just find individuals that are not having this organism, like they're HIV negative, and then on their own, some of them will become HIV positive. You then follow these individuals over time and then look to see if some of these HIV positive individuals come down with AIDS. And so far, the only cause of AIDS that we've detected is the virus HIV. So it's pretty clear that HIV is causing disease, and how we know this is by large epidemiological studies. And that was the same for uh, syphilis and Bartonella henselae. You just study large numbers of individuals who are negative for these organisms, and then you follow them over time. And let's say they come down with the disease, and then you check them, and they're positive for the organism of interest. So that's how we can identify a given organism causing a disease when we cannot use Koch's postulates. What do you do if multiple organisms can cause the same disease signs and symptoms? For example, there's more than one species of virus which causes the human colds. In fact, there's more than one family of viruses. There's 3 virus families that cause the human cold. In Koch's postulate, let's go back. You have to have one microbe causing one disease. The organism must be present in every case of the disease. I guess I didn't read that. And so you can't use Koch's postulates to show that a virus causes the human cold because there's many different viral species that can cause the human cold. What do you do if some species can cause different disease conditions? Like salmonella can cause salmonellosis, but it can also cause typhoid fever. 2 different diseases caused by the same organism. You can't use Koch's postulate on that. And then moral and ethical conditions for human-only diseases, like nobody's ever experimentally injected HIV into a patient and then later looked to see if they became down with AIDS, because AIDS is such a devastating disease. Morally and ethically, you can't study it, at least in humans. And so there are some exceptions to Koch's postulates, and usually what we do is we use vast epidemiological studies to study the disease and what causes the disease. Some terms for classifying infectious diseases. A symptom is a change in a body function that is experienced by a person as a result of a disease, but this symptom is subjective, that it is not observable or measurable by an observer. Like when somebody has a headache, that person is the only one who can know that that person has a headache. You cannot measure a headache. Any question about that? A sign is a change in the body that can be measured or observed as a result of a disease. So instead of being subjective, this is an objective sign. Example, when somebody has a temperature, you can measure it and say, yep, you got a temperature of 38.5 degrees Celsius, which would be something like 102, 103 degrees Fahrenheit. So a sign can be measured by an observer. A symptom cannot. A syndrome is a specific group of signs and symptoms characterize a disease, like the signs and symptoms characteristic of the disease AIDS, the AIDS syndrome. Any question about any of that? I guess I'll leave this here. Classifying different diseases. We have communicable diseases. This is a disease that is spread from one host to another. Examples, measles, tuberculosis, chickenpox. These are all communicable diseases. A contagious disease is a disease that is easily spread from one patient to another. Like COVID-19 is a contagious disease because it's easily spread from one patient to another. Measles, another contagious disease. So which of these three diseases, and it's actually only two because I already told you measles was contagious, which of these communicable diseases is not contagious? Anyone have a guess? Anyone know how easily chickenpox spreads? I guess you guys probably haven't dealt much with chickenpox. Chickenpox spread very easily. In fact, when I was young, even when I was a young adult, parents who had a child with chickenpox would tell their neighbors who had children and said, my son or daughter has chickenpox. I'm throwing a chickenpox party, and if you want your child to be exposed to chickenpox, send him over. And that's because chickenpox was a very mild disease when the child got it. Chickenpox is much more severe disease when an adult gets it. And so chickenpox was actually a chickenpox party was pretty common when I was a child or um, a young adult. And nowadays, because of the chickenpox vaccine, chickenpox parties are a thing of the past. But chickenpox tended to spread fairly easily. Tuberculosis does not spread very easily. So tuberculosis is a communicable disease. It is not a contagious disease. All contagious diseases are communicable, but not all communicable diseases are contagious. Any question about any of that? There are also non-communicable diseases, diseases that are not normally spread from one host to another. For example, somebody who gets botulism or tetanus, they're not going to spread that, at least under normal circumstances. So we call it a non-communicable disease. Yes, it is an infectious disease because a microbe causes it, and you picked up the tetanus bacteria, Clostridium tetani, which causes the disease, but you're not likely to spread it to someone else. There is one exception. Can you think of a way where somebody comes down with the disease tetanus and then it can be spread to another patient? Can anyone think of a way? Nobody? I'll give you extra credit on this one too.
Would it be like blood to blood contact?
A patient has tetanus if it is in the blood, and then he gives blood to another patient and the blood isn't screened, for example, it could spread that way. So go ahead and email me. That is an acceptable answer. That's an unusual answer, and it won't normally happen today because they'll screen the blood and they'll see the
Is
person
it
has tetanus
is
in the.
is tetanus the bacteria that you get from like stepping on a rusty nail?
Yes, that's the endospore you usually get on the rusty nail, not the bacteria itself, but it could be spread that way. Okay, but that's not spreading from one patient to another, but spreading because you get the wound. And what happens is the tetanus has to grow at least partly anaerobically, and so you need a puncture wound to inoculate the endospore inside you, and then the tetanus can start growing inside you. On the open skin, like an open wound, if it gets infected, tetanus is not going to grow because it wants to grow at least partly anaerobically. And in the open air, it won't grow. The way I was thinking about where tetanus could spread is if somebody gets tetanus and then somebody else were to eat him, cannibalism. That would be one exception for how a non-communicable disease could spread patient to patient. And that tells you a good reason why you should not eat other people, because non-communicable diseases can spread that way. Any question about any of that? All right. Let's move on to talking about the occurrence of a disease. To understand the full scope of the disease, we need details about its occurrence. By occurrence, we mean the old and the new cases. The incidence is the fraction of a population that contracts a disease during a specific time. So the incidence is the new cases only. The prevalence is the fraction of the population having a specific disease at a given time. So the prevalence is both the new cases and the old cases. Some diseases, when they're talked about, we usually only talk about the incidence. Like when we're talking about the cases of the flu in 2026, or maybe the winter of 2026-25, we're talking about the incidence, the new cases only. I've never heard of anyone talking about the flu, talking about the prevalence, because the prevalence of individuals in the United States that have come down with the flu would be very close to 100% of the population. So I've never heard the flu talked about in the numbers of prevalence. Any question about any of that? Some diseases, we do talk about the disease more often with the prevalence than we do the incidence. I'm not sure if this is still the case, but with COVID-19, at least during the pandemic, they were always talking about COVID-19 as the prevalence number, the total number of individuals in the United States who came down with COVID-19, meaning both the fraction of the new and the old cases. To show you the incidence and the prevalence, in 2007, the incidence of AIDS, the new patients in the United States that came down with AIDS, was 56,300. But the prevalence of patients in the United States in 2007 with both new and old cases of AIDS were 1,185,000. And these numbers are for 2007. Any question about any of that? All right. When talking about the occurrence of disease, we have different terms talking about how frequently the diseases occur. There's a sporadic disease. This is a disease that occurs only occasionally in a population. Like most people say, typhoid fever in the USA happens sporadically. Every once in a while, we have someone who comes down with typhoid fever. Another example would be malaria. In the United States, malaria is a sporadic disease. Only a few people each year come down with malaria, and usually they come into the United States from outside the United States. And while they're outside the United States, they become infected with malaria and then bring it back to the United States. So, malaria, most people say, is a sporadic disease in the United States. An endemic disease is a disease that is constantly present in a population. The common cold is an endemic disease wherever you are in the world because it constantly is present in the population. You can even come down with the cold in the summer. It's just less likely to happen than in the winter term. Malaria is a disease which is endemic to the tropics. Malaria does not normally spread, at least in the northern United States, because the disease is not normally found in the United States. And then it's spread by mosquitoes biting humans. And in the northern United States, we do not have the species of malaria which can spread malaria. However, in the south of the United States, particularly in the southeast, meaning the Gulf area and the southern Atlantic up to about Washington, D.C., we do have the mosquito species, Anopheles, which is actually a family or a genus that can spread malaria. And every once in a while, someone brings in malaria from the outside the United States, and then a mosquito bites them and spreads the malaria to someone else in the United States. Actually, it was just a year or two ago where I think 3 states in the United States had malaria spreading by the bites of mosquito. One of them was Texas. I think the other was North Carolina. And I don't remember the third. It might have been Florida, but some southern state, southeast state. And what happened was somebody brought in malaria, mosquito bit them, and then it started spreading in the United States. So every once in a while, we can have malaria spread in the United States. And like I said, it happened it was either last year, last summer, or the summer before, maybe the summer before that. What they should do is, if malaria starts spreading, what they should do is just put disinfectant all over the place, trying to kill the mosquitoes that have the malaria. Oops, didn't mean to do that. An epidemic disease is a disease acquired by many hosts in a given short period of time. Like influenza, when it becomes an epidemic, we have a large number of hosts getting the disease over a short period of time. To be a true epidemic, you have to have the incidence of disease greater than normal, statistically greater than normal over a short period of time. We haven't had an epidemic of the flu in the United States since something like 2009. It's been a long time. And usually we have an epidemic of the flu about every 10 years. Now, it's true during the pandemic because people were limiting their spread, wearing masks, other things, that the flu became very rare during the pandemic. But since the end of the pandemic, the flu has actually rebounded, come back, and we've not ever had a epidemic yet. But some year we will, where it'll be significantly greater than usual, and then we call it an epidemic. So this winter, or the closer to the December part of this winter, we had an increased number of the flu, but it was within the normal range. And so it was not an epidemic flu this winter. It was normal cases of the flu. Any question about any of that? A pandemic is where the disease becomes worldwide. So an epidemic is where one region or one continent has a higher than expected incidence of the disease. A pandemic is when it becomes worldwide. For example, COVID-19 was first an epidemic in China, but then it spread to other continents besides Asia, and then it became pandemic. Any question about any of that? All right, let me warn you that when you're talking about a low number of cases, that it's debatable whether disease is sporadic or endemic. For example, let me see if I can remember which one I'm talking about. Tuberculosis. Some people say that tuberculosis is a sporadic disease in the United States. It happens every once in a while at low numbers. Other people say no, tuberculosis is endemic, especially among the homeless population. We always have cases of tuberculosis in the United States. And when you're talking about low numbers of disease, the distinction between an endemic disease and a sporadic disease is minor. It's really dependent upon who the author is and what their definitions are. Any question about any of that? All right. When talking about disease, it's important to talk about the severity or the duration of a disease. So another way of defining the scope of a disease is in terms of its severity or its duration. We have acute diseases. These are diseases where the symptoms develop rapidly, the disease is short-lived, and then the disease goes away. A classic example of an acute disease is the human cold. The disease develops rapidly. First, the patient is well, then they quickly become sick. The disease number of organisms in the patient climbs very rapidly over a very short period of time, but the disease is short-lived. The cold then soon goes away, usually within a week or less. Even the flu can be considered an acute disease. A chronic disease is one that disease develops slowly, persists for a long period of time, and usually gets worse with time. It may get better over a long period of time, but initially it starts slow. You have low symptoms. The symptoms get worse and more severe over time. An example of a chronic disease is mononucleosis, where the disease develops slowly, gets worse with time, persists for a long period of time, and that's why it's a chronic disease. It does start getting better. The disease slowly goes away, usually, and then you're finally cured of it. So that disease is one which is chronic. It does get worse with time, but it does get better over time as well. That isn't always the case. You can have a chronic disease, like in the old days, HIV used to kill a patient once the patient got AIDS. Now, with modern medication, we can keep the patient alive so that the patient is unlikely to die. And it is true that they have a shorter lifespan than normal, but their lifespan is pretty close to normal. It's just I don't know, a few years, maybe 10 years less than normal. But in the early days, everyone who got HIV eventually developed AIDS, and then the patient always died. And so that would be a chronic disease, as well as one like mononucleosis, where it gets worse with time, but then it eventually starts getting better, and then you throw off the disease. There are diseases in between an acute and a chronic disease. You don't need to know about it. The name is subacute disease. You don't need to even know that. Where the symptoms are between an acute and a chronic disease. An example is subacute sclerosing panencephalitis. I'm sure you've never heard of that disease. Any question about acute and chronic diseases? All right. Another way to talk about a disease is to talk about the extent of the host involvement. Infections can be classified according to the extent which the host body is affected. A local infection is where the pathogen is limited to a small region of the body. Example, a boil. Only the skin boil is the infection. Or an abscess. Only the abscess is the infection. A systemic or generalized infection is an infection throughout the body. For example, when a child gets measles, the measles virus will grow almost everywhere in the body. It does grow specifically, I think, in the lungs initially, and then grows in the blood, and the blood spreads it throughout the body, and then it comes back to the lungs, and then it can breathe out. But my point is, it's a central, systemic generalized infection where the disease is growing essentially everywhere in the body. If it's not everywhere in the body, it's close to it. A focal infection is a systemic infection that began as a local infection, such as MRSA that spreads by the blood, and it started from simply a skin lesion, like a boil. A focal infection, a skin boil, that it got worse, it invaded the blood carried it elsewhere in the body, became a systemic infection. Any question about any of that? Let me state that MRSA or MRSA, multi-drug resistant Staphylococcus aureus, is a species of Staphylococcus aureus that is just resistant to many antibiotics, usually to all except for maybe vancomycin. And there's even variations of it that are resistant to vancomycin. So it is a member of Staphylococcus aureus. It's just resistant to many antibiotics. It is a skin disease. It tends to spread from person to person by touch, but the person could touch an object. The bacteria can then be on the object, and then another person can touch the object. So it can spread by fomites, that's the object. It does not tend to spread by the air. And it's a serious disease because usually when someone has a Staphylococcus aureus infection, they will just give usually methicillin. And of course, they're going to be resistant to it. And then they will try the backup drug, which will be another antibiotic, but not vancomycin. And then they'll do a blood, no, not a blood test, a bacteria antibiotic test. And they'll find that MRSA can be resistant to many different antibiotics, usually resistant to every antibiotic tested except for vancomycin. Vancomycin is the fallback for treating MRSA because it's not used generally for anything else because this antibiotic is so harsh on the body. It's really only used for an MRSA infection. But there are some versions, some strains of MRSA that even resistant to vancomycin, and then you have no antibiotic to treat the infection. What do? You have to rely on experimental methods, and that could be something like treating the bacteria with a virus. We'll talk a little bit about that later. Treating the bacteria maybe with um, a bacteriocin. One treatment that's actually commonly used for MRSA, especially if it's a skin infection, is you put honey all over the MRSA, meaning that region of the body, and then maybe just put a gauze on it. And the honey prevents the MRSA from growing too much. too much solutes in the honey, and then the MRSA can't grow. It will dehydrate the bacteria. All right, some terms for the extent of host involvement. Bacteremia, that's the presence of bacteria in the blood. Compare this to septicemia. This is growth of the bacteria in the blood. Usually a clinician will not be testing for growth of the bacteria in the blood. They'll only look for bacteria in the blood. So bacteremia is something used clinically more commonly than septicemia, but you could send the blood out to a microbiology lab and then they could test for septicemia. Toxemia, presence of toxins in the blood. Viremia, presence of viruses in the blood. Any question about any of these terms?
What was septicemia again?
Uh, the ability of the bacteria to grow in the blood. Growth of bacteria in the blood. So you can actually measure the growth of the bacteria in the blood. All right, some more terms about the extent of host involvement. Primary infection is an acute infection that causes the initial illness. A secondary infection is an opportunistic infection that only occurs after the primary infection has weakened the body's defenses. So the primary infection predisposes the individual to getting a second infection that would not happen unless the primary infection was there. A common case of the primary infection is when a child comes down with a cold and then the child gets an ear infection. The ear infection only happens because the child had the primary infection. Another case would be when someone gets one bacteria in their throat and then another bacteria starts growing in the throat, which could be an opportunistic infection, something that was a normal microbiota before the primary infection. We also have subclinical disease, which is an inapparent infection. We had another term for that, an asymptomatic disease. They mean the same thing, where the patient has the disease-carrying organism, but they have no noticeable signs or symptoms. The patient doesn't know they're sick, but they're carrying the disease-carrying organism. This is important for you guys because you guys are young. About 50% of young patients who have COVID-19 have a subclinical disease or an asymptomatic disease. And it's not terribly important for you guys because you're not going to ever notice that you have signs or symptoms. You'll be having COVID-19, but you have no disease signs or symptoms. The trouble is you can carry this organism to someone who then the COVID-19 can infect, and then it can be serious disease, such as your grandparents. And that's the trouble with the subclinical disease. The patient is fine because they have no noticeable signs or symptoms. They don't know they're sick. And then they can carry the disease-carrying organism to someone who it will be much more serious. And I just gave the example of COVID-19 because it's true. About 50% of young people are asymptomatic. And then they can carry this disease to the grandparents. And the more elderly you are, the more severe COVID-19 can be. It's true that some people can carry the polio virus, but they never get disease, signs, or symptoms of polio. Polio, if you don't know, is a very serious disease. If you have disease, signs, and symptoms, you'll become paralyzed. And for a while there, during the war in Syria, Syria had polio show up in Syria. Syria had been free of polio for a large number of years before the fighting started. And then obviously the medical care fell down People stopped being vaccinated for polio, and the disease came back. I think recently Syria has thrown off polio, meaning they vaccinated enough people, and so um, there's been no known cases of polio disease in Syria. However, they're not calling the country disease free yet. You have to be something like 10 years without disease before they'll be called disease free. And I think polio is only being spread in two countries in the world, Afghanistan and Pakistan. And all the other countries in the world have no signs or symptoms of polio. Much of the world is polio free, but not all the world is polio free. In talking about the severity or the duration of disease, it's important to realize that the disease could be a latent disease, and then there's herd immunity. I think I'm going to stop here and say that I will continue here next time. Any questions about any of that? All right. You don't need to show up in the lab unless you have questions or you want to hear questions. I will be in the lab for 15 minutes to answer any questions at 6:30. All right. Bye.